Hey guys, this is me, Miss Eisenhower. I'm going to do your lecture on osmosis and diffusion, which is cellular transport. I'm going to do it this way because it's really not that hard of a concept. So I thought it would be easier if we just kind of do it this way. And you're going to see me kind of looking to the side some and then turning my laptop some back and forth because I'm taping on my laptop and looking on my desktop at the PowerPoint here. So that way I can use the board behind me because I think the board behind me will help give you a good visual if we do it this way. So go ahead and open up the PowerPoint that you have in Google Classroom that I sent you. And you can split screen them if you want to. And that way, if you split screen them, you can be looking at both of them at the same time. You could be seeing me, and then you could be seeing your PowerPoint at the same time. So it'll kind of make a little bit more sense to you. So we're going to go ahead and get started on cell transport on things like osmosis and diffusion, passive and active transport. So we're going to start first with the cell membrane. We're going to talk more about the cell membrane when we talk about actual parts of the cell, which is our next lesson that kind of goes along with this lesson. But for right now, what you need to know about the cell membrane, is the cell membrane is a, let me erase that right there, rewrite it down a little bit. This is going to be inside the cell, and this is going to be outside the cell up here, okay? And when I talk in terms of the cell membrane, I say that I have a phospholipid bilayer. I have a bilayer here. Here's the actual bilayer. I've got a layer on the top and I've got a layer on the bottom. And I have a phospholipid bilayer because my phospholipid bilayer is made of phosphate heads and lipid tails. Here's my phosphate heads. These are lipid tails. And if you'll remember back, lipids are nonpolar molecules. They are hydrophobic, and hydrophobic molecules do not like water. They don't like to mix with them. So that's why your phospholipid bilayer arranges itself like this. They keep those lipid hydrophobic tails on the inside so they don't get exposed to any kind of water on the inside and the outside of the cell out there. So this cell membrane is what we call semi-permeable, and that means it lets some things through but not everything through, and that's necessary for cell regulation because we've got to get some things in, we've got to get water in, we've got to get nutrients in and things like that, but we've got to get waste out. But we can't just let a flow of everything comes in or our cell's going to rupture and burst, and we can't just allow everything to flow out or our cell's going to shrivel up and die. So we've got it. This is our regulator. This cell membrane is what's going to regulate what goes in and what goes out. We say it's selectively permeable because it doesn't let everything in and out. It only lets certain things in and out. And this is what controls the regulation of substances that come into and out of the cell. When we talk in terms of transport, whether this is going to be passive or active transport, we have to talk about the concentration gradient. The concentration gradient is the concentration of a substance against the concentration of another substance. So we're going to be comparing one region into another region down there. And we're going to talk about the gradient in terms of a higher concentration to a lower concentration. If I would look at it right now, these are my sodium chloride molecules. Sodium chloride is regular old table salt. If I look at it just right here like this, I have a higher concentration of sodium chloride up here than I do inside the cell down here. And my concentration gradient would be this difference from this region outside the cell to the region inside the cell right here. So this movement, this cellular movement, is going to take on one of two forms. It's either going to be considered passive or it's going to be considered active. If it's passive transport, that means that no energy was needed for the transport of those molecules. If it's active transport, that means that you actively are using energy, specifically the ATP molecule, which is adenosine triphosphate, which we'll talk more about when we talk about photosynthesis and cell respiration. We're using energy. We're using ATP. When I have passive transport, I'm going from a concentration of high to a concentration of low. That's passive transport. Just think of it as in you are at the top of a hill and you roll down. Well, when you roll down the hill, you really don't have to put any energy in it. So there's no energy and you just kind of falling and rolling down the hill. But when you've got to run back up the hill, you've got to use energy to run back up that hill. You've got to get your way back up there. Or think of it as in if you are, if you're tubing, if you've ever been tubing and you've went up to the mountains or somewhere and you're tubing and you're at the top of the mountain and they take you up there in the car and you get in that tube, you just sit there. You sit in that tube and you just kind of lazily sit there and you go down the mountain. You're going from high up in the mountain, just sitting in that tube and riding it down the mountain. But if they tell you you have to swim your tube back up to the top of the mountain, you're going to use a lot of energy swimming that inner tube back up where you just came from, trying to go back up that high mountain. So think of it that way. You're going from high to low. You're just sitting in the inner tube going from high to low. You're passively not using any energy. But when you go from low to high, you're going against the concentration gradient. You're going to have to use energy. And that is, you're taking that inner tube from the low at the bottom of the mountain. You got to swim it back up to the top of the mountain up there. You've got to use energy to get that inner tube back up to the top of the mountain. So that's a good way to remember between passive and active transport. Passive, no energy. You're just sitting there going down the mountain. Active, you're actively swimming it back up, so you have to use some energy. There's a couple different passive and active. When we talk in terms of passive, there's three passive. There's osmosis, diffusion, and facilitated diffusion. And then there's, when we talk in terms of active, there's going to be molecular and bulk, and we're going to break all of those down and look at them. When I talk about the very first one, I talk about diffusion. And diffusion is simply the movement of molecules from a concentration of high to low. That's it. Diffusion moves them from high to low. Eventually, diffusion will stop when they reach equilibrium, and equilibrium is when they're the same on both sides. They're equal. There's an equal concentration of molecules on this side as an equal concentration on that side. Think of diffusion as if you have some perfume and you have perfume in a vial, and not a spray bottle, because a spray bottle you'd be using something to help, using some energy trying to get that out. But if I just simply have perfume in a vial that has a little top on it, and I take and I pull the top off of that perfume, well, eventually you're going to start to smell the perfume. Well, the reason why that is because those perfume molecules are highly concentrated inside that jar, and when I take the top off, I let them go from an area where they're highly concentrated to where they're out into the room to where there's very low concentration. So they're trying to equal out the equal amount from inside that tightly packed molecules inside the jar out into the room. And eventually they'll stop moving because they'll flow from an area of high to low. There's three things that are going to affect diffusion, three primary things that are going to affect diffusion. The material, if it's a solid, liquid, or gas, that will be three things that'll affect it. The size of the molecule, larger molecules have a much more difficult time getting through. Small molecules like oxygen, water, carbon dioxide, they can slip through here. They can slip between the phosphate heads and the lipid tails and kind of work their way through. But larger molecules, they can't. And we're going to talk about that in just a second. So carbon dioxide, oxygen, and water for the most part, but we're going to talk something about water. There's special things for water to get through too, because water's a little bit bigger than carbon dioxide and water, so it needs kind of a helper sometimes. But then the last thing that will affect it is temperature, because if we increase our temperature, remember, we're going to increase our kinetic energy, and when we increase kinetic energy, that means we've increased the movement of the molecules, and they're going to be bouncing all around and moving faster, so they can get through faster when we increase the temperature. The next slide that you have just kind of shows you how they work their way through oxygen, CO2, and water. And like I said, we're going to talk about a special property of water here in just a second. Facilitated diffusion. Facilitated diffusion is another one they'll always move with. They move with and down the concentration gradient. They're the same thing. They're interchangeable in there. So if I move with the concentration gradient, I'm moving down the concentration gradient because I'm always going from high to low with passive transport. Now, the thing about facilitated diffusion is exactly that. You have a facilitator. We have something that's going to facilitate this. Now, you're going to get channels that are going to come in here. I always have students that put facilitated diffusion in the active transport side. It is not active transport, okay? Because whether it's active or passive has to do with the concentration gradient, whether you're going from high to low or low to high. The use of this channel is not what makes it active or passive. So I always have students say, oh, this molecule is too big. They're using, it has to use a channel to go across, so it must be going against. No, that's not it. Whether it's active or passive only has to do with whether the concentration is high to low, which is passive, or low to high, which is active. Okay? So facilitated is just simply that. We are going to use something that is going to facilitate our transport across. And these are going to be proteins, and they're going to be special proteins that are embedded in the cell membrane. And these are going to be transport proteins. You already know what proteins are. So now we're going to start to figure out what we're going to do with these proteins. And I told you before, proteins are, they're it when it comes to us in biology. They run the show. So there's 3 types of facilitated diffusion in terms of proteins. There's 3 types of proteins that facilitated diffusion uses. The first one, let me erase this so I can move this over. All right, the first one is going to be just a simple channel. And this simple channel is simply going to be just like that. It's going to be a channel that is specific only for one particular molecule. That's it. It's only going to let that one molecule through. Okay? And in this case, it's only going to let a hydroxide through. It's going to let the hydroxide through all the way through here and have no problem. Hydroxide can slip back and forth all it wants to through this particular channel. These guys cannot. Because remember, when we talk about proteins, we say form means function. So these protein channels are only going to be specific for this hydroxide. That's it. Nothing else is getting through there. Okay? But that's just a simple channel protein. Nothing else goes on with the channel protein right there. Now, we can have, and you're just going to have to bear with me because somebody lost the piece for this kit that we have, so I have to use this one for the same thing. We can have something called gated channel proteins. And gated channel proteins, what they do is they will allow only one certain molecule through because they're proteins, and proteins are specific. But the thing about gated is that you have to have some type of signal molecule has to attach. So what that means is there has to be some type of signal molecule that attaches somewhere on this gate right here to allow, if this is down here, has to signal on here to allow this gate to open up and allow this molecule to come through. It's a gated channel. Think of a gate, like if somebody has a gate on their property, and some people have a gate and they have a remote control to it, so when they pull up, they hit a remote control and it opens their gate. That's what a gated channel does. This particular molecule has to come and attach. It's like the remote control. It has to come and attach to open up the gate and allow the molecule to come through. And again, it's specific because proteins are specific. Okay? And then the last one is a carrier protein. This is a carrier protein, right? And carrier proteins, let me get caught up with you on my PowerPoint here. Let me go to carrier. Carrier proteins right here, they do exactly that. They actually will carry, we say, a molecule across. what happens with these guys is that the protein will temporarily open up. It will change shape. The molecule will come up and it will enter. And then the carrier protein will open up on the other side. And when it opens up on the other side, it lets the molecule out. The key point with these guys is that they temporarily change shape and they temporarily have a binding site for whatever molecule they're carrying across. It will temporarily change shape, open up. It will temporarily bind with the molecule. Then it will open up and it will let that molecule go through there again. Okay? So let me go back because I need to tell you about a specific channel protein. Remember, a channel protein is simply just that. It's a channel that just lets one particular thing through. There is a specific channel protein that's called an aquaporin. And an aquaporin is just that, aquaporin. Aqua, water, pour, an opening. An aquaporin is specific to let water molecules through. It's a channel protein that just allows these water molecules to go on through. Nothing else can come through because proteins are specific, and these are specifically aquaporins that only allow water molecules to come through. Okay? So 3 types: channel, carrier, and gated. Okay? Channel is going to be specifically just a channel that allows a specific molecule to go through. Aquaporins are specific channel proteins. They just have an opening and they let that specific molecule go through. Then we have carrier. Carrier actually temporarily change shape and temporarily allow that molecule to bind in there, carrying it to the other side. Then we have gated channel, and the gated channel, you have to have a molecule, a signaling molecule, to actually open up the gate and let the molecule actually come through by opening up that gate. Okay? So those are the 3 in terms of facilitated diffusion. But again, even though I'm using a protein, one of those 3 proteins, I am not going from a concentration of low to high. I'm still going from a concentration of high to low, so it's still passive transport. Okay? And then there's simply just some pictures in there in the PowerPoint that I put in there for you so that you could kind of see and get a little better visual of it. Okay? All right. Now, there is a specific type of passive transport, and it's called osmosis. Osmosis is simply the movement of water, a diffusion of water across a semipermeable membrane, whether it be with the help of an aquaporin or simply just slipping through. Okay, so osmosis is passive transport, and it's simply the diffusion of water across a semipermeable membrane. So water is going to go from a concentration of high. Water is going to go from a concentration of high up here to low down here until we finally balance out. Okay? This is going to be the balance of water. Now, when we talk about osmosis, osmosis is going to keep happening until you get equilibrium. The key to this being is that when you get to where it's equal on both sides, then you'll have an equal movement of molecules of water, keeping you equal. Okay? So it'll keep trading like that. Right now, if I would have it like this, my concentration of water would be much higher up here and lower down here. So these water molecules would move here until I got equal. Okay? When I get equal with my water molecules, I'm going to say that this is an isotonic solution. The concentration of this solution is the same as the concentration of the solution here. Now, that's what I'm going to say. When I talk in terms of isotonic, hypotonic, and hypertonic, I'm talking about the concentration of my solutes. Okay? The case that I'm using here is salt water. Salt is going to be my solute. Remember, water is my solvent. It's my universal solvent. And this is going to be, here's some sodium chloride, here's some sodium chloride, and here's some sodium chloride. Okay? All right. I have to get my concentration the same on both sides. All right? Once it's the same on both sides, then I have what we call an isotonic solution. And when I have an isotonic solution, that means I'm going to get the same amount of water movement inside and out. It's going to be equal. And that's what I say, these water molecules are going to be equal because I have the same concentration of molecules up here as I do down here. All right. I can have a hypotonic solution. If I have a hypotonic solution, that means the concentration of the solute is lower outside of the cell than it is inside of the cell. Okay? So, if I would want to make a hypotonic solution here, it would have to look like this. These are my sodium chlorides. My magnets aren't strong enough. These are my sodium chlorides, which is my salt. Okay? So, all these sodium chlorides right here, there's 1, 2, 3, 4 here. There's only 2 on this side over here. Get this other stuff down so you don't get confused. I've got 4 down here. I only have 2 out here. This is inside my cell. This is outside of my cell. So in this case, the solution on the outside of the cell is hypotonic to the solution on the inside of the cell. Okay? It is in this hypotonic solution that you're going to have more water outside the cell, right? Because if I have a lower concentration of solutes, I'm going to have more of a concentration of water molecules in there. So in this case, the water would be moving in because it wants to try to balance out that concentration that we have here. Okay? So here is when water moves into the cell. This hypotonic solutions are what gives plants their turgor pressure. And turgor pressure for plants is the pressure that holds the plant upright. That's what we call turgor pressure. It keeps the plant from wilting. When you have a lot of water loss from a plant, their turgor pressure goes really low, and that's when they start to wilt. But if their plant's turgor pressure is high, then that's when the plant is standing upright and it's rigid. The cell walls are rigid and the cells are full of water in there. So plants actually like a little bit of a hypotonic solution because that's what helps them stay upright, and it helps them avoid the gravity pulling them down if their turgor pressure is up and full and upright. It is also called osmotic pressure in animals. So it's turgor pressure in plants, osmotic pressure in animals. Now, if you've got a little bit of salt in your body and in your blood, you know that from things like sweat and tears and things like that, that you've got some salt in your blood. So if you were to take and you were to put an animal cell in, and you were to take and put it in distilled water, then what would happen is that the water would rush in because it would be like this and it would try to balance this salt concentration out. The water would rush in and it would cause what we call cytolysis. Remember I told you to make sure you learned what lyse was? Lyse is what? Lyse means to kill or to break apart. Cyto means cell, so we're going to have cytolysis here. is you're going to have a lot of water to rush in, and you're going to rupture this cell. You're going to burst it. You're going to burst this cell membrane. Plants are able to do it because plants not only have a cell membrane around them, this is just a cell membrane. Plants have a cell membrane around them, but then plants also have a rigid cell wall. And that rigid cell wall, which by the way, if you've forgotten, is made from the cellulose, carbohydrate cellulose, structural carbohydrate cellulose. That rigid cell wall is what helps keep the plant from bursting. And we're gonna look at that a little bit more. You'll look at it in lab. Okay, so plants are fine with hypertonic solutions. Us, not so much. That's why when you go to the hospital, they give you saline, and that saline IV has a little bit of salt in it. They don't give you distilled water IVs. They give you saline because they know if they give you just distilled water IVs, the water would rush in and it would burst your cells in there. Hypertonic solution. All right, hypertonic, the concentration of the solute is higher outside of the cell. So this time, we're hypertonic. Our concentration is much higher outside of the cell than inside of the cell. So we got a hypertonic situation right here where my concentration of solutes is higher out there. So here, when we have this happen, this is when the water wants to balance everything out. So the water's going to leave to balance out this higher concentration that I have up there. Now, when this happens in plants, it's called plasmolysis, and you'll actually look at that too. You're gonna put some salt water onto some plants leaves, and you're gonna see what's gonna happen. The water's gonna leave, and it's gonna go to the salt water. And when it goes to the salt water, the plant, you're gonna actually physically watch the cell membrane, and what'll happen with that is that the cell membrane starts to pull away from the cell wall from the water loss, and the things become very concentrated in here, and your cell membrane will draw up from the water leaving the cell because the concentration is much higher out here. It's called plasmolysis in plant cells, and when it happens in animal cells, it's called crenation, and it causes the animal cell to shrivel up and die from crenation on there. And then the next slide I give you shows you exactly what happens if you put an animal cell in different osmotic pressures. Cells placed in distilled water just shows you they'll swell and burst, and then it'll show you cells placed in a concentrated salt solution. Crenation will happen, and they'll shrivel up and die. And then the next one also just kind of shows you some visuals of what goes on. The osmotic pressure in plant cells. You'll see where the cell will stiffen, but generally retain its shape. Now, you can put the concentration so much that you can rupture the cell walls, but usually the cell walls are a good barrier there, and the cell just kind of expands some and it stays full. But if you place it in salt water and they're in a hypotonic solution, that water's going to leave to balance out to balance out where my solutes are in there. Okay. So that's passive transport. Passive transport's not using any energy. I'm still going from concentrations of high to low in here. The key about this is that I want you to assume, unless you're told, water is what's going to be doing the moving. Okay? Unless you're told that something is permeable to that membrane, and you will have some problems like that, but you will be told. If not, you assume water is the only thing that can cross that membrane, so water has to be the thing that's going to balance your concentration out in your numbers. Okay? All right, so all this has been high to low, high to low, high to low, high to low. Okay? So let's look at active transport. Active transport is simply going the other way. We're going to be using energy, and we're going to be using energy to go from an area of low concentration to an area of high concentration. This does not have to do with osmosis or anything. This is active transport. where we're actively pumping something against the concentration gradient. We go from low to high, and we use proteins. Now, this is where students get messed up. Because we use proteins, they say, oh, facilitated diffusion has got to be active transport. No, we're going to use proteins, but we're using them to pump, act as a pump, to pump molecules from a concentration of low to high. Okay? And the one, well, I'll get some molecular. We're simply going against the concentration gradient. Okay? There's two types of active transport. There's molecular and there's bulk. Now, the molecular example is going to take these ions and pump them from a concentration of low to high. And one of the big ones is the sodium-potassium pump. That's a primary example of pumping sodium and potassium ions back and forth from a low concentration to a high concentration. That's all we have to know for in here. That sodium-potassium pump has to do with things like cardiac function and nerve conduction. You don't have to know how many sodium pump across per how many sodium per potassium. That's stuff that you'll learn in AP Biology. Those of you that go on to take AP Biology. For in here, you just have to know the example is the sodium-potassium pump is the example of molecular active transport. And it's simply pumping sodium and potassium ions from a concentration of low to high. We're going back up the mountain again, so we're using energy. That was molecular. And then there's bulk transport, okay? Bulk transport can come in two ways. It can come in endocytosis and it can come in exocytosis. Endo means within. So endocytosis means you're taking things into the cell. And there's two parts to endocytosis. There's what we call cell eating and cell drinking. There's phagocytosis, which is cell eating, and that's the cell taking in larger molecules. And by they take in larger molecules, they engulf them. That's things like when a cell, like you might have maybe a macrophage or a phagocyte, which are part of the immune system, and they'll go and they'll engulf things like bacteria and other large molecules to fight off disease and fight off bacteria. But that's large molecules or phagocytosis. And then penocytosis is cell drinking, and that's small molecules. That's mainly the cell taking in things like extracellular fluid and things like that. It's really small fluids and very small ions and things like that. So we say phagocytosis is the very large engulfing of things like macrophages that will engulf things like bacteria. And then penocytosis is cell drinking down there. It's taking in very small molecules and taking in extracellular fluids and things like that. And then exocytosis is when things are going to exit the cell. That's when we're going to get rid of waste. We're going to do the reverse of the endocytosis. We're going to get rid of things outside of the cell. And if you'll look at your next slide right there, when we talk about cell organelles, when we talk about cells right after this unit and we talk about cells, we're going to talk about cell organelles. You have things in there called vesicles and vacuoles. Vesicles are just a smaller type of vacuole. And these guys are responsible for endo and exocytosis. These vesicles and vacuoles have a lipid bilayer around them. And it's important that they have a lipid bilayer around them because if you look at your PowerPoint right there, you'll see what's going on. I can show you my screen in case you can't see. Right there. Right there. Hopefully you can. Well, that's not going to work out, guys. I tried. If you look there, it's important that these vesicles are made of the same thing. Because what happens with that is, let me take all this down. What happens with it is that you have the cell membrane, okay? And these vesicles are surrounded by a phospholipid bilayer too. So what happens is if this is my cell right here, and inside my cell I have these little vesicles like this. Well, what happens with them is in endocytosis, these vesicles will move up here to the lipid bilayer. And what they do is they fuse. They'll actually move up here and they'll fuse with the cell membrane. So these guys will kind of open up and fuse like this. And this is what you'll get. You'll kind of get this little dipped in area. Well, what happens is when the molecule moves in here, then these guys will pinch off. These little vesicles will pinch off. This will go back to being a little vesicle in here, and then this vesicle will move inside the cell, and it's got whatever it engulfed in it. Then the exact same thing's going to happen after some cellular processes are going to take place. There's things in these vesicles like digestive enzymes and things like that. Once these cellular processes take place, then these vesicles will move back out to the cell membrane, and then they'll temporarily bond with the cell membrane, and then they'll release the things outside of the cell. So that's how we get things into and out of the cell in terms of bulk transport. And this whole thing of bulk transport does require energy. It's a type of active transport, okay? So when we talk in terms of cell transport, we talk about passive and active. If we talk about passive, we're specifically talking about diffusion, facilitated diffusion, and osmosis, which is the specific diffusion of water. All of those things are going to go from a concentration of high to low. If we talk about active transport, we're talking about things that actively need the input of energy to do it, because you're moving things from a concentration of low to high, or you're physically needing energy to allow these vesicles to bring things into the cell through endocytosis or out of the cell through exocytosis. Okay, so that's the lecture for passive and active transport. And I'm going to stop this and let you watch it now. And then I'm going to do a different video to work some problems with you, some transport problems with you on osmosis and diffusion. And that way you can know what you're doing by working these problems. But in order to do that, I have to access my document camera. So I have to change some things up a little bit. So let me know if you have questions on this, and hopefully you can understand it. Bye.